The initial tblastn search we performed in Part 2 helped define the search region for the putative ortholog within the genomic scaffold of the target genome, yakuba. The next step in our analysis is to determine the approximate coordinates of each coding exon, or CDS, of Rheb-PA in yakuba. The approximate coordinates of each CDS can be determined by aligning each CDS of the gene in D. melanogaster against the search region of our target genome. To do this, we will utilize the Gene Record Finder and BLAST. FYI, because the BLAST algorithm does not take the positions of potential splice sites within a complete protein sequence into account when it generates an alignment, BLAST often extends or truncates the alignment beyond the coding exon boundary and into the intron. To ameliorate this issue, the Pathways Project Annotation Protocol recommends mapping each coding exon separately to determine their approximate locations, and then further refine the coding exon boundaries by searching for compatible splice donor and acceptor sites through visual inspection using the genome browser. In addition to comparing a query sequence against a collection of subject sequences in a database, the NCBI BLAST web service allows us to compare two or more sequences against each other using the program BL2seq, short for BLAST2 sequences. To map the amino acid sequences of each D. melanogaster CDS against the yakuba scaffold, BLAST must translate the entire yakuba scaffold sequence in all 6 reading frames, 3 reading frames of the positive strand and 3 reading frames of the negative strand, and then compare each conceptual translation against each CDS sequence from D. melanogaster. Thus, we will conduct a tblastn search using the CDS as the query and the nucleotide sequence of the scaffold as the subject. Here we will perform 5 tblastn searches, using each D. melanogaster Rheb CDS sequence as the query and the accession number NC_052530 we identified for the yakuba scaffold in Part 2 as the database or subject sequence. Scroll down to the CDS usage map under the Polypeptide Details tab. Since Rheb has 5 CDSs, we will need to run 5 different tblastn searches, one for each CDS. Let's start with CDS1. To view the protein sequence for CDS1, select row 1, flybase ID 1_9824_0. Copy the protein sequence, including the header, shown in the pop-up window. The size of amino acids column indicates how many amino acids are in each CDS. For example, CDS1 is 16 amino acids long. To set up the tblastn search, navigate to the NCBI BLAST website. Click on the tblastn image under the Web BLAST section. Paste the sequence for CDS1 into the Enter Query Sequence text box. Select the Align 2 or more sequences checkbox. Note NCBI BLAST is using the BL2seq program. In the Enter Subject Sequence text box, enter the accession number for the yakuba scaffold. In Part 2, we found the best collinear set of alignments to Rheb-PA in yakuba was at 19,150,809 to 19,151,699 on the NC_052530 scaffold. We can now use those coordinates to narrow down the search region of the yakuba scaffold by limiting the subrange of the NC_052530 scaffold to roughly 100,000 base pairs on each side of the collinear set of alignments. The subject subrange 19,051,000 to 19,252,000 is determined by subtracting 100,000 from the smaller coordinate, adding 100,000 to the larger coordinate, and then rounding each to the nearest thousandth. Based on our analysis in Part 2.3, limit the subject subrange by entering from 19,051,000 to 19,252,000. FYI, the average novel contains about 400,000 characters, and the NC_052530 scaffold in yakuba is 30,730,773 base pairs long. Since BLAST must translate all 6 reading frames, the search is roughly equivalent to reading 462 novels worth of A's, T's, C's, and G's to find a few pages of important information. Here, we limited the search region to 19,051,000 to 19,252,000, approximately 100,000 base pairs upstream and downstream of the range we found in Part 2 to increase the statistical power of our search and help filter out spurious matches. The default NCBI BLAST parameters are optimized for searching the query sequence against a large collection of sequences in a database. When we are using BLAST to compare only two sequences against each other, we need to change some of the algorithm parameters because the default parameters could potentially mask the conserved regions of the coding exon. Click on the plus icon next to Algorithm Parameters to expand the section. In the Scoring Parameters section, change the Compositional Adjustments field to No Adjustment. In the Filters and Masking section, uncheck the Low Complexity Regions checkbox in the Filter field. Select the checkbox next to Show Results in a New Window, then click the BLAST button. Here is a summary of the tblastn search as it is displayed on the results page. The D-melanogaster CDS1 query is 16 amino acids in length, and the program searched 201,000 translated nucleotides of the NC_052530 scaffold in D yakuba to find a match for the 16 amino acids in CDS1 of D-melanogaster. FYI, you may find it helpful to download your BLAST results in case you need to refer to them later. Click on Download All and then Text in the drop-down menu. The tblastn results show a single match with an e-value of 1e to the minus 6 and a sequence identity of 93.75% to CDS1. Click on the Alignments tab to view the corresponding tblastn alignment. The query coordinates show that the alignment covers all 16 amino acids of CDS1. Note, we can find the length in amino acids of CDS1 in D-melanogaster using the gene record finder, or at the top of the tblastn search results. The subject coordinates correspond to the region within the NC_052530 scaffold of D yakuba, 19,150,809 to 19,150,856, that shows sequence similarity to CDS1 from D-melanogaster when it is translated in the third reading frame of the positive strand, frame +3. Hence, we can place CDS1 of Rheb at approximately NC_052530 19,150,809 to 19,150,856 in D yakuba. We can apply this same procedure to place the other four CDSs on the NC_052530 scaffold of D yakuba. See Appendix A for instructions on how to combine or batch multiple sequences. Copy the CDS2 Rheb 2_9824_2 sequence along with the header from the gene record finder. Return to the tblastn web browser and delete the CDS1 sequence from the Enter Query Sequence text box. Paste the CDS2 sequence in the text box. Leave everything else the same as we had for CDS1. Click on the BLAST button to run the tblastn search. Click on the Alignments tab to view the corresponding tblastn alignment. Repeat steps 15 through 19 to blast the remaining CDSs. Here is a summary of the tblastn CDS by CDS search results. Examination of the subject ranges for the tblastn alignments of the 5 CDSs of Rheb in D yakuba shows that they are collinear. CDSs 1 through 5 are placed on the positive strand, and the subject subranges for the CDSs are in ascending order. Consequently, the CDS by CDS search results support the hypothesis that the putative or probable ortholog of Rheb-PA is located at approximately 19,150,809 to 19,151,702 on the NC_052530 scaffold of the D yakuba genome assembly.